The second function of the innate immune system is to identify what is foreign and differentiate it from what belongs in the body, and then basically remove the foreign material by activating specialized white blood cells. Now, when we think about the white blood cells, there is no one type of white blood cell that makes up the innate side of the immune system. It includes T cells of a variety of types, including what are called natural killer cells, neutrophils, macrophages, and a type of innate cell called a dendritic cell. The dendritic cell gets its name because it looks a lot like a neuron in that it has these projections of membrane coming off of it that look like dendrites. The general function of these cells is to, again, identify what material is foreign and then essentially eat it and digest it or destroy it. So remember, the term that we use for this eating process is phagocytosis. And once the foreign material is inside of the white blood cell, it can be destroyed, it can be taken apart. These cells will be targeting primarily infectious living material like bacteria and other microbes, damaged cells, cells that are no longer functioning and need to be removed, and also cancer cells, cells that are dividing abnormally. The innate immune system can take care of removing and destroying all of those different types of cells. Now, on this slide, we can see a diagram again of the cells that make up the innate immune system. Now, we don't need to worry about all of the detail on this slide, but it does remind us that a lot of these cells come from primarily the bone marrow. And what they're showing us at the top is that there is what is referred to as a hematopoietic stem cell in the bone marrow. Now, if you're not familiar with the term stem cell, what it describes is a cell that, when it divides, it doesn't produce two identical daughter cells. It produces one daughter cell that will go on to mature and become a specific type of cell. The other daughter cell will be a stem cell. So in other words, stem cells are unique in that they continually replenish themselves while also producing cells that will go on to mature and become some specific type of cell. In the diagram, they're showing us that the hematopoietic stem cell can then branch off into two lines of innate immune cells. One is called the lymphoid, and the other is called the myeloid. So these two cells, the gray one and the orange one, represent the progenitor of each one of those lines. These are not true stem cells, these progenitor cells, but they give rise to all of the other types of cells in each one of these lines, the lymphoid line and the myeloid line. Now, the lymphoid line includes both T cells and B cells. The T or T lymphocyte cell and the B or B lymphocyte cell. The myeloid progenitor gives rise to the cells that we think about in the blood that are the white blood cells. For example, the neutrophil, the monocyte, and this is the dendritic cell. The myeloid progenitor also gives rise to white blood cells that live in the tissue. Remember, in all tissues in the body, not just the bloodstream, we find at least one type of immune cell. So macrophages, again, dendritic cells. These are found both in tissues and in the lymph nodes themselves. The next function of the innate immune system is to recruit other immune cells. So when one of the cells in the innate immune system discovers and identifies something as being foreign, something as needing to be destroyed, it's not only going to do the phagocytosis process to destroy that thing, but it's also going to send out signals that will bring other immune cells to the area. These are chemicals that it is secreting, and these are what are called cytokines. Cytokines are actually a type of protein. They are a relatively small protein, and they're involved in communication between cells, particularly between cells of, again, the immune system. Cytokines get released or secreted by one immune cell and then bind to and act on another immune cell. So, all the cells we've mentioned so far in the innate immune system, macrophages, T lymphocytes, B lymphocytes, mast cells we haven't quite mentioned yet, even cells that are not specifically part of the immune system can respond to cytokines. And these include things like endothelial cells. Those are the cells that line blood vessels, and fibroblasts. This is a connective tissue type of cell. So there are many different cells in the innate immune system that can secrete cytokines and respond to cytokines. And there are non-immune cells that can respond to cytokines. Now, what exactly makes up the cytokine family? Well, there's a whole range of chemicals that are considered cytokines, and you may have heard of some of these. There are the interferons, the interleukins, there are chemicals called chemokines, lymphokines, things like tumor necrosis factor. It's a whole range of chemicals. And these are chemicals that serve to recruit other cells into this area of the body that is undergoing an immune reaction. The next function of the innate immune system is activation of the complement cascade. Now, we're not going to be able to talk about the complement cascade in detail, but you should know a few key points about it. The complement system is actually composed of over 25 different proteins whose job it is to assist or complement, if you will, the action of antibodies in destroying bacteria. What happens with the complement cascade is that as individual proteins in this system become activated, they go on and activate the next protein in the system, which then activates the next protein, and so on and so on. And that's why it's referred to as a cascade. Remember, after an immune response occurs, and immune cells have done the process of phagocytosis, and they've secreted cytokines and recruited other cells into the area, what ends up happening once that immune response is finished is that there's a fair amount of debris that needs to be cleaned up. And this is one of the systems in place to help clean up. The complement cascade will get rid of all of the antigen antibody complexes. We haven't talked about antibodies yet, but we will. The complement cascade is responsible for dilating local blood vessels, which allows immune cells that are in the bloodstream to come out of the bloodstream and into the tissue where the immune response is occurring and help to, again, clean up the debris that's in the area. The complement system is um, aiding in or contributing to the process of inflammation in that area. So part of the redness and the warmth and the swelling that we think of during inflammation, during an immune response, comes from the proteins in the complement system. Something else to know about the complement cascade is that the very first protein in the cascade is activated as soon as an antibody binds to the foreign material, a part or a portion of the foreign material at least, that we refer to as an antigen. Again, the first protein will then activate a second protein, which will activate a third, and then a fourth, and so on, until the whole cascade of proteins becomes activated. This is the system, this complement cascade, which will help clean up the immune reaction. It will help clean up what's left behind. It can also help in the process of breaking down what's left behind, some of these microbial cells, for example. And the complement cascade will continue on by implementing inflammation in recruiting other cells to that area. Now, this is a very busy slide. I put this slide in not for you to write anything down in your notes, but just so that you can see a drawing of how complex this cascade is. And it really is very, very complex. What they're showing us is a cell, a bacterial cell, for example, right here. It's labeled as a pathogen. And then over here, we can see a host cell, an immune cell. And you can see that there are antibodies involved here. You can see one here and here and here. And yes, antibodies exist on the surface of certain immune cells. You can see all of these different C structures labeled C2, C3, C4, C6, C8. They're all shown in blue here. These are the individual complement proteins, and they're showing us one by one how they all get activated and what they do, how they bind to not only the pathogen cell, but also to the immune cell, the host's immune cell, and cause this whole cascade of things to happen. It's a very complex system and a very important system in the innate immune response. The last function in the innate immune system is to activate the other system, the adaptive immune system. And this is a process that's referred to as antigen presentation. Now, I mentioned the antigen a minute ago as being a portion of the foreign material. By definition, what an antigen is a macromolecule that can elicit an immune response. So it's not the entire bacterial cell. It's some piece of that bacterial cell that functions as the antigen, that alerts the immune system that this thing is foreign. Antigens are oftentimes proteins, but they can also be other types of macromolecules, including polysaccharides, lipoproteins, and glycoproteins. Antigens tend to be relatively large molecules. They are macromolecules. And an antigen may have multiple sites on it that are considered immunogenic. So we give those sites that occur on an antigen a name. We call those epitopes. So an epitope is a place on an antigen that helps to elicit an immune response. Antigens can enter the body from the environment, Antigens can also come from within the body. Remember, the immune system will not only recognize an invading microbe as being foreign and needing to be destroyed, but it can also recognize cellular material inside the body that needs to be destroyed. Cells that are not functioning, cells that are damaged, cells that are cancerous, and so on. Unfortunately, in some disease conditions, this system goes awry a little bit, correct? You've all heard of autoimmune disease, and it is possible for the innate immune system to begin to see antigens on normal, healthy, functioning cells. We don't fully understand the pathophysiology of autoimmune disease, but we do know that in an autoimmune condition, the normal innate immune response is functioning abnormally in that it is identifying as foreign epitopes on antigens that are a normal part of the body. And unfortunately, because the system sees them as foreign, the system goes about destroying them. And that's why you develop disease processes like lupus, like rheumatoid arthritis, hemolytic anemias, and so on. So, part of the job of the innate immune system is to identify epitopes on antigens as foreign and to respond to them. And the last component of that response is to present that antigen to the other side, if you will, of the immune system, to present it and then activate the adaptive immune system. On this slide, you can see a table of the epitopes for the HPV, or the human papillomavirus. So there's nothing for you to learn on this table. I just wanted to show you that the human papillomavirus has been very well studied, and in fact, we know now how to manufacture a vaccine against this virus. This is a vaccine that is now routinely given to young men and women to help prevent human papillomavirus infection. And what you're looking at here is a list of the locations on the protein antigens on this virus. So peptide number, this refers to specific locations in the protein that are considered antigenic, that have regions or epitopes on them that will stimulate an immune response. You can see that epitopes get classified as being either major or minor. There's only one major epitope on this particular antigen for the human papillomavirus. What you're looking at here is referred to as the epitope sequence. Now, this is not a sequence of bases because we're not talking about DNA or RNA. This is a sequence of amino acids. Every amino acid has a couple of abbreviations associated with it. You've seen some of these abbreviations in the genetic code table. So PHE for phenylalanine and VAL for valine. But each amino acid also has a single letter abbreviation, a single capital letter abbreviation. And that's what you're looking at here. A string of letters that tells us the order of amino acids in this particular epitope. These are the epitopes that we then target the vaccine against, because we know that these particular regions on the antigenic protein will elicit an immune response.